So, Lab Services has been working with a customer to measure an active pharmaceutical ingredient or an API in an over-the-counter medicine. We were trying to measure this because when these products are cleaned by the manufacturer, they need to know how much of their product is still left in the cleaning solution after that cleaning process is done. So you need to be able to get to very low concentration levels. So we were trying to see if we could use spectroscopy to solve this issue for them. And so we were inspired by this customer and wanted to expand our knowledge base. So we decided to test a different over-the-counter medicine known as ZzzQuil, which is a nighttime sleep aid. And the API or active ingredient in ZzzQuil is diphenhydramine HCl. We took the ZzzQuil solutions as they are, and that has a certain concentration. And then we did a serial dilution of the ZzzQuil in water and tried to determine the limit of detection using UV-VIS absorption spectroscopy. So we were able to determine that we could get to a very low concentration level of the active ingredient and identify the peaks associated with this ingredient in the UV-VIS absorption spectrum. In the absorption spectrum, we were able to identify the key features that correspond to the diphenhydramine. Once we can identify that, then we can monitor how that peak in the absorption spectrum decreases with decreasing concentration. So from that, we can develop a concentration curve and predict the concentrations based off the absorbance values.